Okay, well, welcome everyone to Paediatric Teaching for this week, and good afternoon to you all. Today and next week, I wanted to talk about genetics in paediatrics, and it's something we don't learn much about, but it's very relevant to many aspects of pediatrics and so many different aspects of medicine. And I wanted to try to explain some of the basics of genetics, something about the history of genetics. And I think by understanding the history of genetics and the basics, then we'll understand a lot about how it applies to clinical practice. Yeah. So this is the first talk. I'm going to cover quite a lot of ground, and I thought we'd cover the history of medical genetics. And it's very interesting. I think it goes back about 160 years. And by understanding the history of medical genetics, I think you can really get a good understanding of what tests are these days done for different genetic conditions and how our understanding of genetics progressively over time has led to a greater understanding of the genetic basis of diseases. Part of that is to understand how to study chromosomes and how that has evolved from just looking at karyotypes or even looking at patterns of diseases, karyotypes, and then more modern technology to study chromosomes. And not so much you need to understand that, but understand the basis for which we understand things about genetic diseases. I'll talk a bit about cytogenetics because it's very important. That's a one method of studying chromosomes, cytogenetics, and it has enabled an understanding of a whole lot of different diseases that previously weren't known about. I'll talk a bit about chromosomes in cancer. I mean, again, for those who are doing the part one examination this year, understanding chromosomes in cancer is a frequent question in the exams. I'll talk a bit about biochemical genetics because that really applies a lot to children and especially inborn errors of metabolism. We'll say a bit about immunogenetics because we use it every day. Every time you do a blood cross match, that relates to immunogenetics. And so the earliest understandings of immunogenetics were about blood groups and that sort of thing. But there's been more understanding of different types of immunogenetics that help us understand different diseases. I'll speak a bit about molecular genetics, a more modern method of studying chromosomes and studying genetic diseases. And then we'll talk a bit about newborn screening, which is in many countries something that is done for all babies at the time of birth and then to try to identify genetic diseases. Even though it's not done in all countries, it's still something that's good to understand the types of conditions that can be identified early by newborn screening. And there's a couple in PNG that would be useful to have some method of newborn screening. I'll mention those. Genetics is not just about making diagnoses of rare conditions, but there are many different ways to treat genetic disorders these days, from very simple ways to more complex ways. And then lastly today, I just want to go through the way genetic information is encoded within chromosomes and is translated into proteins. Now, most of you will have studied that at a medical student level and probably for your part one examinations, but that part of it will be just a bit of revision, but I hope that it will be useful. So we'll get underway. The earliest geneticist as such was a monk in Austria, and you may have heard about this. I read this book over summer, and it's called The Friar Who Grew Peas, and many of you will have heard of Gregor Mendel. He was a monk and then an abbot in an Augustine monastery in Austria, and he discovered the laws of inheritance long before it was chromosomes were known about. And he did this by experimenting on garden peas over about 7 years in the sort of 1850s to 1860s. So he came a bit after Charles Darwin, but he understood very much that there was genetic variation in all living creatures, and he grew peas and other vegetables in the garden, and he understood the genetic basis of this, and he did this by many experiments over, as I said, 7 years. He worked out that if you cross a tall pea plant with a short pea plant, then the result is you get all tall peas. But if you then allow that tall pea plant to self-fertilize, then 1 in every 4 of those pea plants, or roughly 1 in 4, will be a short one. And you might recognize already the Mendelian pattern of inheritance that is being displayed here. But this is being a tall pea plant was obviously an autosomal dominant condition. It was dominant over the shorter version of the pea plant, but it could come out because there could be, with self-fertilization, there could be a pea that had that was short without a tall pea gene. And this is the you might have heard of a Punnett square, which is it's an interesting term because it's a genetic term, but it relates to a punnet of vegetables or a punnet of peas in Gregor Mendel's experiments. And it just shows that if you cross a tall a dominant with a dominant, you'll get a dominant. If you cross a dominant with a recessive, you'll still get a dominant. If you cross a dominant with a recessive, you'll get a dominant. But if you cross a recessive with a recessive, then you'll get a recessive phenotype. And so the chance of any dominant phenotype, if you cross a heterozygous tall and short pea, is 1 in 4. The chance of a dominant phenotype is 3 in 4, as Mendel found, and the chance of finding the recessive phenotype, that is a short pea, is 1 in 4. This was his first major experiment. That these were his actual numbers there. You can see that they almost came out to 3 to 1, which is exactly right, isn't it? Should be, statistically, it should be 3 to 1, 3 talls or dominant phenotypes to 1 short one. That was in the 1850s, and then about 30 years later, the first human chromosomes were seen in tumor cells. So already it was clear that when cancer cells were being examined, it was clear that there were chromosomes within the cells, and that was later on found to be inherently present in all human cells and all cells, not just in cancer cells. It wasn't known at that stage that cancer had a genetic basis in any way, but the first human chromosomes were seen in cancer cells. It was about that time, about 10 years later, around about 1900, that the theory that chromosomes carry determinants of hereditary and development was made, and that harked back, I suppose, to Mendelian type of inheritance. They proposed that there's a chromosomal theory for Mendelian, Gregor Mendelian's descriptions of inheritance. And then it's in the next 10 years, some other genetic types of genetic diseases were identified, and we'll talk a bit more about these because they're inborn errors of metabolism. And it's only in 1908, in the actual second edition of that British medical journal called The Lancet, that inborn errors of metabolism were described, and they described several inborn errors of metabolism, which you might have heard about: alkaptonuria, pentazuria, cystinuria, and albinism. You've probably heard of some of those inborn errors of metabolism, and you may have even seen the phenotypes of some of those conditions. This was the first method for studying chromosomes, and it's worth knowing about. Here the chromosomes are distributed as, 22 pairs of autosomes, 1 pair of sex chromosomes. This is called Giemsa banding, and it enables certain and grey bands to be seen on the chromosomes. This is a karyotype. This is an old-fashioned karyotype, And it was possible by using this method to detect chromosomal breakage, if there was a breach or a break in the chromosome, chromosomal loss or duplication or translocation, which I'll describe, where there's an additional chromosome translocated from one chromosome to another, or inverted segments of the chromosome. This was the earliest type of chromosomal analysis using GIM sustaining, the G banding of chromosomes. This is a schematic picture of it, but it shows that all karyotypes are arranged in a similar way. 22 pairs of autosomes, 2 paired sex chromosomes, so either XX or XY, and I don't know whether you would be able to identify, but whether this is a male or a female karyotype. But for those who have had some familiarity with studying chromosomes, you'll be able to tell that this is a male karyotype because the X chromosome is longer and the Y chromosome is shorter. So if it was a female karyotype, this sex chromosome would have 2 long chromosomes as a sex chromosome, but because it's a male, 1 long and 1 short. The short one is the Y chromosome. I wanted to go back a bit to, well, come forward a little bit, if you like, to what happened next. So the earliest discoveries of genetics were made in the 1850s and 1860s, right up to about 1900. And then after that, there was I would say, a dark period in medical genetics where very unfortunate events happened to the development of genetics. And that occurred in many countries, and it was called the era of eugenics. And this was because people realized that genetics was the basis of all characteristics. They thought that it might be possible to breed out bad genes, and for even bad genes were racial. They were intellectual disability, or they were considered bad genes if a person had a physical disability, or even if they were poor, because that was considered to be something related to your intellectual level. And so in many countries, there was attempts by governments to improve their gene pool and prevent the dissemination of bad genes in future generations. This is called eugenics. And it was in the early 1900s, it was considered the science of human betterment. In many countries, including the USA and of course Nazi Germany, it was the justification for the Holocaust. But surprisingly, in many other countries, there were eugenics programs, and the eugenics program in the United States went for about 40 years, and in Sweden even went for longer. It was a shock to me to realize that, and many even medical journals, even reputable medical journals, were involved in this eugenics period. Even when the American Society of Human Genetics was established, like that is now a very reputable society. Many of those, the former directors of the society, were part of the Eugenics Society. And it's now recognized, of course, that eugenics is a form of abuse and a gross violation of human rights. And it took 40 or 50 years for people to realize that. But that's certainly the way most people would think today, that eugenics is something that's very bad and a dark past of part of the history of genetics. There were much more positive things to come. In the 1950s, this concept of cytogenetics was found, and that enabled, instead of just a chromosomal break or a translocation or a loss of a chromosome or an addition of a chromosome to be identified, then single gene defects were able to be identified using what we call cytogenetics, which is the analysis of genes within a cell, cytogenetics. And one of the first discoveries was that of the gene defects causing hemoglobinopathies like sickle cell disease and thalassemia. They were very early discoveries, and very important to know that thalassemia was a genetic disorder. Before the 1950s, it wasn't clear that that was the case. in 1956, the normal number of chromosomes was identified as being 46, so 22 autosomes, one pair of sex chromosomes, a total of 46 chromosomes. And then soon after that, it was then found about different disorders where you have an additional chromosome. So, of course, Down syndrome, and in a number of other trisomies like trisomy 13 and trisomy 18, that they were identified in the 1950s and 1960s. There's another abnormal number of sex chromosomes, or chromosomes were identified as Turner syndrome. You probably heard of Turner syndrome, that occurs usually almost always in females, where they have short stature, they have a number of other cardiac abnormalities and a characteristic facial appearance, and also another sex chromosome abnormality was Klinefelter syndrome, which is XXY, where it's a male phenotype with very tall stature, some other different characteristics, infertility and very tall stature. So these changes in chromosome numbers and structure, and even basic single gene defects, were identified in the 1950s and 1960s. As I said, some of the main discoveries in that time, and I'll try to go through them because you need to understand the terminology. Mosaicism means when there are two or more chromosomally different cell lines in the same individual. So for some genetic defects, like with Down syndrome, for example, there are some children who've got Down syndrome who are what they call mosaics. That is, not all of their cells within their body will have an additional chromosome 21. Some will have a normal number, like just two 21 chromosomes, and some cells will have three, and that's just called mosaicism, and it relates in a way to the time in embryo development when the abnormality occurred. And if the abnormality occurs after the initial first phases of cellular duplication, then some of the chromosomes will be normal, and those that occur afterwards, those cells that develop from the earliest embryonic cells that contain the translocation, will be affected. And usually when children have a mosaic Down syndrome, they have a more milder form of Down syndrome. So there is a spectrum of patients who have Down syndrome, and there's a spectrum also of, I mentioned this a condition of Turner syndrome, where children, girls mostly, have short stature. They have sometimes a coarctation of the aorta, sometimes they have a webbed neck, and they have a characteristic faces, and they may have some learning difficulties. They're usually some level of cognitive impairment, but they're usually able to function pretty well, like children with Down syndrome. But sometimes there'll be a mosaic Turner syndrome, where some of the cells will be XX, as in the normal female karyotype, and some of the cells will be XO, which is the Turner syndrome karyotype. And so there'll often be milder forms of Turner syndrome as well. In the other term to know is like structural changes in chromosomes, and some of those will be like additional arms of a chromosome. They're often what we call translocations of chromosomes, not always, but sometimes translocations, where or deletions, where there might be one part of the chromosome, not just one gene, but one whole part of the chromosome is deleted. That's a structural change, an addition or a deletion, chromosomal deletion. Translocations are where extra material from one chromosome is transferred to another chromosome, and that occurs in some forms of Down syndrome. For example, there's a chromosomal abnormality you might have heard about in Down syndrome called the Robertsonian translocation, which is the translocation of one part of chromosome 21 onto chromosome 14. So they may appear to have just two chromosome 21s, but there's a translocation of the additional the trisomy chromosome onto their chromosome 14, so-called Robinsonian translocation. And the children are still the same. They still have all the characteristic features of Down syndrome. It's just that when you look at the karyotype, it looks different. It's called the Robinsonian translocation. After I showed you Giemsa banding to start with, and that was the earliest way in which chromosomes could be identified and genetic diseases identified, chromosomal abnormalities identified. In the 1970s came a more modern way and a more detailed way of looking at chromosomes, and this is called FISH, or fluorescent in situ hybridization. And that it's color banded instead of just being grey and black, and that enabled the detection of chromosomal breakage and chromosomal loss and duplication and translocation or inverted segments at a much more I would say microscopic level, or a much greater level of detail. And so because of that, many other diseases were identified that had these other more minor chromosomal abnormalities. So this is a form of cytogenetics, and in the 70s and 80s, after FISH was first introduced, a whole lot of other conditions, as I said, like Langer-Geerden syndrome, you might have heard of that. That's a condition where children have short stature and they have bony hyperostosis, really thickened bones in different parts or lumps in their bones, and they have some intellectual disability as well. And Prader-Willi syndrome, which, as you probably know, children have morbid obesity, but when they're born, they're actually often very hypotonic. They're quite floppy babies, and then they later on develop an insatiable appetite and put on very large amounts of weight and have some intellectual disability as well. DeGeorge syndrome, that you might have heard about, a condition that is often linked to congenital heart disease, but also linked to aplasia of the thymus. And so children have a T-cell immune deficiency. And Beckwith-Wiedemann syndrome, again, a sort of a macrosomic type syndrome where children are often have what we call hemihypertrophy. They're macrosomic at birth, and they may have a larger body habitus, and they're a bit more likely to get omphaloceles, as you probably know. So all of these conditions were identifiable by a fluorescent in situ hybridization, a more complex way of looking at genetic diseases. So from this, microdeletions and duplications and other very small abnormalities were able to be seen, and also the what was identified in cancer cells, the under or overexpression of certain genes that led to that cancer, the so-called proto-oncogenes, and I'll explain what they are in a moment. Proto-oncogenes are genes we all have, and they stimulate cell division. So we normally have to have cell division. They inhibit cell differentiation, and they inhibit cell death. So it's helpful to have proto-oncogenes. They have a normal function in human development and maintenance of tissues and organs. The problem is when you get mutations in the proto-oncogenes, they turn into oncogenes, and that leads to the unregulated production of the same proteins and massive amounts of cell division, no cell differentiation. So differentiation means one cell can differentiate into another type of cell, can transform into a different type of cell for a different function. Well, if there's unregulated cell division and there's no cellular differentiation, then that means the same type of cell is being produced multiple times, like thousands and millions of times, and that is what leads to cancer. And the other thing, if you have an oncogene, then it may lead to an inhibition of cell death. There's a natural process of cell death, but if you have unregulated cell division, no cellular differentiation, and no cell death, then there'll be just a continuous growth of the same cells. That is a cancer. Virtually all malignant cells carry some chromosomal abnormalities. And this was identified as early as the 1960s. And one of the earliest chromosomal abnormalities identified was the lesion called the Philadelphia chromosome. And that was identified in patients who've got chronic myeloid leukemia, usually adults. But of course, the Philadelphia chromosome is part of the chromosomal changes that occur in some forms of acute leukemia as well. And it's a translocation between the long arms of chromosome 9 and 22. You'll remember what a translocation is when one part of the chromosome is translocated to the other part. And when the cell divides, then the translocation is passed on, as well as the other normal chromosomes. So you then have an extra piece of chromosome in all the subsequent cell divisions. And that leads to uncontrolled activity of the gene responsible for cell division, and you get uncontrolled growth of myeloid cells in chronic myeloid leukemia. And so the Philadelphia chromosome is now considered one of the risk factors, the higher risk factors for some forms of acute leukemia in children. If a child's got the Philadelphia chromosome, then that puts them into a higher risk group. There's been similar translocations identified as the PH1, the Philadelphia chromosome, in other leukemias and lymphomas involving different chromosomes and cell growth generators. The Philadelphia chromosome is a large chromosomal change, but most of the proto-oncogenes and the oncogenes that are much smaller mutations, maybe one or two gene defects in these proto-oncogenes. We all have tumor suppressing genes, it's important to know. And of course, if you get a genetic mutation in a tumor suppressing gene, then you're likely to have unregulated cell division because they won't suppress the growth of those tumor cells. So these discoveries of the chromosomes in cancer and the genetic abnormalities that occur in cancer have been very important in understanding a whole range of cancers, including many pediatric cancers. And this process I showed you earlier about fish, that fluorescent in situ hybridization method, showed microdeletions. Remember, you can see down to a very small level to microdeletions, 1 or 2 gene defects, showed microdeletions in Wilms tumor and retinoblastoma. And now we know that there is, a genetic basis for some forms of retinoblastoma and some forms of Wilms tumor, but all Wilms tumors and retinoblastomas carry some gene defects, some microdeletions, most of which have in most children with Wilms tumor or retinoblastoma have occurred after embryonic development. But there are some forms of retinoblastoma, bilateral retinoblastoma, that are inherited forms of retinoblastoma. I wanted to speak a bit about biochemical genetics. We've talked about cytogenetics and chromosomal abnormalities. I want to speak a bit about biochemical genetics because it's very important for pediatrics. As I mentioned earlier, in 1909, so over 100 years ago, this first concept of inborn errors of metabolism was identified, published in The Lancet, and it was about there were 4 different inborn errors of metabolism identified at that time. But it wasn't until the 1950s that was a number of other enzyme deficiencies were identified, and these might be familiar to you. One is glycogen storage disease, not G6PD, but glucose 6-phosphatase deficiency in the glycogen storage diseases, and a disease called phenylketonuria. Phenylketonuria it certainly used to be the most common cause of intellectual disability in adult males. That's what it was, and it used to in the early 1950s, it was identified that this was probably due to phenylalanine hydroxylase deficiency and a gene defect in the phenylalanine hydroxylase gene that encodes for that protein, which is the enzyme, and PKU was the first disease identified that could be could be found on newborn screening, and simple dietary modification, a form of dietary modification, could lead to the prevention of intellectual disability in those patients, a little bit like congenital hypothyroidism, which I'll talk a bit about later. And then after that, a whole lot of different inborn errors of metabolism were identified, including these conditions called mucopolysaccharidosis, where you get mucopolysaccharide. It's like a sugar deposition in tissues and thickening of all tissues. You probably heard of the mucopolysaccharidosis, Hurlers, Hunters, Sanfilippo's. There's a whole range. There's 5 different types of mucopolysaccharidoses, and they were identified as being defects in enzyme production in the early 60s. Then in the 70s and 80s, as more advances occurred, the urea cycle enzyme defects were identified, and even disorders of mineral metabolism like Wilson's disease, that condition that causes liver dysfunction, especially, and some other things, as a copper deficiency, and a condition that you will have heard about called hemochromatosis, where children get, people, adults as well, get deposition of iron in their liver and heart and other places, hemochromatosis. That's relatively common, actually, hemochromatosis as a cause of liver dysfunction. So all of these different types of biochemical abnormalities were identified, and that formed the basis of this biochemical genetics, which is another branch of genetics which we sort of need to understand. I mentioned at the start that I described very briefly immunogenetics, and that's really just identifying antigen and antibody markers, and the antigens are on blood cells, so white blood cells or red blood cells. And as I said, the earliest types were the identification of the ABO blood groups, and around about that same time, the RH blood groups as well. So when we cross-match blood, we are using a test that derives from the earliest efforts in immunogenetics. And so it was around about that time in 1940 that the RH system was described. And then soon afterwards, this condition, erythroblastosis fetalis, which we understand these days to be hemolytic disease of the newborn, that was found to be due to RH incompatibility between the mother and the child. And then very soon after that, anti-D was introduced, and that led to a major fall in the 60s of 1960s of this condition called kernicterus, which is bilirubin encephalopathy, where very high levels of unconjugated bilirubin lead to brain injury. And so now it's very, very rare to have children with bilirubin encephalopathy or kernicterus these days, because of the anti-D that mothers can get if they're Rh negative, and because of the identification of the immunogenetics and blood group incompatibility. So later on, there were abnormalities or genetic markers identified on lymphocytes and other blood cells. So we talked about red cells, but there was also genetic markers identified on B cell and T cell lymphocytes, and that some of those were found to be related to certain diseases. So there are certain immunodeficiencies that are related to abnormalities on B and T lymphocytes that lead to a severe form of, it's called severe combined immunodeficiency, or SCID, which is those children look quite like they've got fairly advanced HIV, but it has a genetic basis to it rather than related to the virus. And then there are the HLA system, which is the human leukocyte antigen system, that was found also on lymphocytes and other immune cells. And now, of course, we use that to do certain things. Firstly, if a patient's having a bone marrow transplant, then they have to be HLA matched. if there are certain diseases that relate to different types of HLA risk or HLA genetic abnormalities, particularly ankylosing spondylitis. You probably would have heard about that. And HLA-B27 positive, they're more likely to get this rheumatological condition called ankylosing spondylitis. And it's now an aid in the investigation of certain autoimmune diseases like type 1 diabetes. It's thought to have probably a genetic basis, although it also has an environmental basis, a combination of the two, almost certainly. With the advances in genetic testing, there were new methods identified. And I won't go through these. You've probably heard about many of them, but this is like a timeline of when different genetic tests were available. So you've probably heard of restriction enzymes, or sometimes called RFLPs, restriction fragment length polymorphism, which is a way of cutting up a gene and identifying different types of abnormalities. And then this thing called the southern blot and the western blot were identified, and gel electrophoresis, where the chromosomes, the genetic material is distributed on a gel, and you can see the genes much easier. The first cloning of human genes occurred in the 1970s, so that was really important because it was able to clone the alpha and beta chains of hemoglobin, which has become important in understanding the changes that occur in thalassemia, alpha and beta thalassemia. These things I mentioned before, the restriction fragment length polymorphisms and gene mapping, that just identified that there's major different variations in genes within cells, and not all of them lead to disease. Many genetic variations just don't have any phenotypic abnormality. The polymerase chain reaction was identified in the 1980s, and then I guess not the final, but one of the big advances was the total sequencing of the human genome, which means that all of the genes in the human body have been mapped in some way. And now there's techniques called whole exome sequencing that enable you to be able to map the entire genome. Because of all these changes, a whole lot of other diseases were identified. For example, chronic granulomatous disease in the 80s, and that's a inherited disease of immunodeficiency, where children are much more likely to get certain bacterial infections. They're more likely to get Pseudomonas and Neisseria, meningitis, and Staph aureus infections. You've heard of Duchenne's muscular dystrophy, which is the most common cause of muscular dystrophy or inherited muscle weakness, almost always in boys because it's an X-linked condition, and of course cystic fibrosis, which it causes bronchiectasis and suppurative lung disease, and also pancreatic inflammation and pancreatic insufficiency. These from molecular genetics, it was also identified that there were some familial cancer syndromes where people within families are more likely to be at risk of certain a multitude of different cancers. There's a sort of endocrine cancer syndrome, family syndrome, and there's chromosomal breakage syndromes where, for example, X-rays are more likely to damage people's chromosomes if they have this condition called Leigh-Fermani syndrome, and they're more at risk of some forms of cancer. Again, familial cancer syndromes were identified using molecular genetics. I simply talked briefly about the newborn screening, and the Guthrie card. You probably heard about the Guthrie card. That was brought in the 1960s, and of course we use the Guthrie card these days for a whole range of different things, including the identifying it's a dried blood spot. So we use dried blood spots for HIV, PCR, and TB testing sometimes. So it just uses filter paper, as you know. The first disease for newborn screening, or the first two diseases were really phenylketonuria, which I mentioned before, and that led to successful dietary manipulation and prevention of a lot of intellectual disability in boys affected by PKU. And then hypothyroidism was the other one. The identification early in life of babies with hypothyroidism led to thyroxine replacement, and therefore the prevention of intellectual disability and all the consequences of hypothyroidism. These days, there's a whole lot of other diseases that are identified using newborn screening. In some countries, sickle cell disease, thalassemia, even cystic fibrosis is identified in some countries. And then there are now other forms of prenatal diagnosis that use a combination of these techniques. So sometimes using FISH, this fluorescent in situ hybridization, mapping or analysis of the chromosomes, sometimes using enzyme analysis, like looking for alpha fetoprotein being elevated in embryos that have a neural tube defect. It's been useful. This sort of thing has been very useful in identifying, in epidemiological studies, for identifying that certain genetic abnormalities increase the risk of certain diseases, like even folic acid deficiency increases the risk of neural tube defects. That was based on a whole series of prenatal diagnostic tests. There's a newer technique called chorionic villus sampling. Now, it's been around for about 40 years, I suppose, but that amniocentesis is obviously the sampling of amniotic fluid, and doing these, in some places, doing a range of tests to identify abnormalities in people at risk. Now, I wanted to talk a bit about treatment of genetic diseases, and that's because sometimes you might imagine that genetic problems are simply they can be diagnosed, but they often can't be treated. And that's true for some genetic diseases, but for many, there actually has been a treatment, and they've all been based on the understanding of a particular disease and the genetic basis of that disease, and usually the protein that is abnormal. And so I mentioned before phenylketonuria, and dietary restriction of phenylalanine can lead to normal intellectual function or near normal intellectual function. And then another one, galactosemia, which you've probably heard about, where babies are at risk of E. coli septicemia and cataracts and a few other things. Well, if you restrict galactose, then they don't get that deposition. They don't get the changes of children with galactosemia. So the identification of these diseases early can lead to, simple measures that can result in much better outcome. We've already discussed enzyme replacement for congenital hypothyroidism. Factor VIII therapy in haemophilia, that's an obvious one. Children with haemophilia, they have a deficiency of factor VIII. It has a genetic basis, X-linked mostly. And if they get replacement of that factor, either with factor VIII concentrate or with fresh frozen plasma, or even with whole blood, then they not be cured of their haemophilia, but their haemophilia can be treated. And it's really only by understanding these protein deficiencies that occur on a genetic basis that we've been able to do that. There's other types of treatments, like there's chelation therapy. Chelation is where you give a substance that binds a toxic substance within the blood. So with Wilson's disease, of course, it's copper. With hemochromatosis, it's iron. Remember, the chelating agents also apply to children who've got thalassemia, who have lots of blood transfusions. They need chelation therapy. Then there are some diseases for which institute of exchange transfusions or dialysis can lead to the can lead to a correction of the enzyme deficiency. And then some conditions just are vitamin responsive. There's a deficiency of a certain vitamin or a cofactor, and the replacement of that vitamin can lead to a much better outcome. So the point is that relatively simple treatments have been applied in the last 60 or 70 years to treat some genetic conditions. These days, there are even more like gene replacement or direct enzyme replacement therapy that can be given for some disease. This is like the things that have happened in the 21st century. And even these days, some children with thalassemia or other inborn errors of metabolism are having bone marrow transplants to correct their disease. We won't go into all that, but just to know there's a wide spectrum of different treatments for certain genetic conditions, and it's worth understanding. Okay, I think we might stop there because we've talked about um, the history of medical genetics, and we've talked about a lot of different things. I'm the next step in this two-part series, if you like, is talking about. I'm going to stop there, but the next step is to talk about the mechanics of genetic disorders, and so this next week we'll talk about um, how genes within cells encode proteins and how that happens. I'm sure, again, you've studied that at a part one level. We'll talk about ways to identify genetic disorders. We'll talk about different mutations. A lot of this will be revision and adding to the things that I've talked about today. I want to cover Mendelian inheritance patterns. We've talked a bit about that, about autosomal dominant conditions, but we'll talk about all the different types of conditions: autosomal dominant, recessive, X-linked, etc. We'll talk a bit about mitochondrial inheritance next week, because there are genes not only in the nucleus of a cell, but also in the mitochondria. And we'll talk a bit about gene regulation and epigenetics and some common genetic disorders in PNG and what the genetic basis for those are. So we'll cover those things, I think, next week, but we'll just stop there, and I'm happy to answer any questions that you might have, or we'll continue on next week.